Yeah, good afternoon, Prof. Cheng.
Hello. Hi, good afternoon.
Okay, maybe host can turn off the music, maybe. Good afternoon, Prof. Cheng.
Hi, Rosa.
Yeah, Prof. Cheng. It's quite a long time ago
Yes,
since we have met in 2018. 18, yeah, 18.
18, huh?
Yeah, 18 Swarna Dipa Physiology quiz. Yeah. Okay. Actually, maybe the dean already here is preparing, and for some student, actually we already informed. Maybe some of them still try to join our Zoom meeting. Okay, and okay, this time also some professor, our lecturer, and also student already join this meeting. There is yes, some teacher also, and student are all in from all study program from Faculty of Medicine of Andalas University. Currently, we have 26 study program. Okay, and also yes, some. Okay, maybe yeah, Italy now already 1 p.m. Maybe the dean still preparing. So, and also there is for the MC today, there is Dr. Mutia Lailani, Prof. Cheng.
Hello.
Hello, Dr. Mutia. Dr. Mutia, or usually called by Dr. Lani, also from Department of Physiology.
Hello.
Department of Physiology.
Professor Chang.
A little bit earlier than before, because maybe around 12 we already have Zuhur pray. So usually at 1 p.m. we can start the activity. Okay. Mulai lagi, Pak. Mulai deh. Okay, Prof. Cheng, because the time already 1 p.m., maybe we can start our event, the lecture. Maybe Dr. Lani, ya, please continue.
Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, Professor Chang. Good afternoon, our Honorable Dean, Professor Akriwadi, and Dr. Rosa Sukmarita. It's so nice to meet you here today in our special events, which is Public Lecture and Visiting Professor with Professor Cheng Wiming from Malaysia. And here I would like to inform you that this event will be present in our YouTube channel as well in Faculty of Medicine of Andalas University. So you may also enjoy these events in our YouTube channel as well. And without any further ado, we will present Dr. Rosa Sukmarita as our moderator today. And before that, we would like to open our events today by our Dean, Mr. Afriwadi, specialty in sports medicines. For Mr. Afriwadi, please welcome.
Sarung. Ya, Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
I'm sorry. Good afternoon, ladies and gentlemen, Honorable Vice Dean, Professor and Lecturer of the Faculty of Medicine, Andalas University. Our honorable speakers, Professor Cheng Hui Ming from University Malaysia. And to all the students from the study program, Faculty of Medicine, Universitas Andalas. I'm pleased to welcome you to the public lecture, Visiting Professor by Professor Cheng Hui Ming. This public lecture is part of the Visiting Professor program, the first time it has been held at the Faculty of Medicine. I would like to thank Professor Cheng for this willingness to become a visiting professor at the Faculty of Medicine, Andalas University. I hope this public lecture can motivate students to increase the enthusiasm for learning and improve their academic ability. Thank you for the all. Thank you, Prof. Cheng. Assalamualaikum warahmatullahi wabarakatuh.
Thank you for our Honorable Dean, Mr. Afriwadi. And we are very lucky today to have Professor Cheng Hui Ming, which has been known as an expert in physiology worldwide. And he has more than 17 books, if I'm not mistaken, about physiology, which is the most interesting one is a question-based physiology, which is very outstanding. And without any further ado, our events today will be moderated by Mrs. Rosa Sukmarita. Dr. Rosa will be our moderators today for the topic Physiology of Salt by Professor Cheng Hui Ming. And the floor is yours, Rosa.
Thank you,
Okay.
Mutia.
Okay. Thank you, Dr. Mutia Leilani. Okay, Assalamualaikum warahmatullahi wabarakatuh. Okay, maybe good afternoon, ladies. The Honorable Dean, Vice Dean, Lecturer of Faculty of Medicine, Andalas University, and of course, the Honorable Speaker, Professor Cheng Hui Ming from the University Malaya, Malaysia, and of course, to all students and participants of today's public lecture. Okay, maybe first of all, I would like to introduce a brief biography from the Professor Cheng Hui Ming. So Professor Cheng Hui Ming has taught physiology at the University Malaya in Kuala Lumpur since 1986. And Professor has an interest in understanding how students think, learn, and in helping them unmask misconception in physiology. And then his books include Thinking Through Physiology and Defining Physiology. And of course, Prof. Cheng is also the initiator of the annual International Physiology Quiz. And luckily, our Faculty of Medicine, visited by Professor Cheng Hui Ming in 2018 in Swarnadipa Physiology Quiz. So actually, we already have known with Prof. Cheng Hui Ming previously, and also with Dr. Arcadius. Okay, maybe for this before the public lecture begins, the first session is presentation from Professor Cheng Hui Ming, and then we have a discussion part. So all the participants can ask the question to the Professor Cheng Hui Ming. Okay, now please allow me to welcome Professor Cheng Hui Ming to deliver his presentation. Prof. Cheng, time is yours.
Thank you, Rosa, for the introduction. Just checking that the sound system is okay.
Sound system is okay,
Okay.
Prof.
Yeah, as Rosa said, my connection with Andalas very much was through the International Physiology Quiz that I chair each year, and also through my friendship with your with the late Professor Arcadius, and who also invited me to initiate. At that time, the idea was to have a regional quiz just for all the medical schools in Sumatra. Yeah, so we did it once in 2018, and perhaps the idea can be resurrected again. Yeah, so it's my privilege to talk to the students who are learning physiology this afternoon, and my topic will be it will be a general big picture of the importance of salt, and in particular we are thinking about salt. It will be sodium chloride, and my focus will actually be narrowed down to basically the physiology of sodium cation and its accompanying anion, which is chloride. Yeah. So let me just start to share screen, and then we will take it from there. Okay. Can the slides be seen?
Yes, we can see the slide, Prof.
Yeah. So so I think I got the right picture from Google, and this must be your, I assume, your new campus.
Yeah, new campus.
It's very impressive.
Okay.
So yeah, so I saw it in Google, and I also saw it when the dean was talking. So the focus this afternoon is on sodium homeostasis. So I tried to modify the title to hopefully engage the audience and just call this by the common name salt. Now, if you're wondering what this cat is doing here and with the balance here, this will become very clear soon. And this is because we're talking about a major cation. So this is where the cat, the major cation, and the balance here is basically coming from the concept of homeostasis. So we're talking about actually the homeostasis or sodium balance. So that's why this cat and representing the major cation sodium, and the concept of maintaining normal sodium balance in the body. So the details will come on in a short while. Okay, so let's move on to the first slide. So this will be the three subtopic which I'll deal with briefly. One is the importance of this cation transmembrane gradient, which is obviously related to sodium. And I think this may be a common knowledge to all students, but I hope to show you that this same gradient is exploited in many physiological events in different organs. Just one simple gradient. And then we'll talk about briefly control of sodium concentration, the word in bold here, and how the concentration control is actually separate from what we mean when we talk about sodium balance. This last one is referring more to the total body sodium in the body. And these are all cat pictures that was presented, I think last year in Sri Lanka. So let's move to the first one, the sodium transmembrane gradient. So this is somewhere behind my house. So so normally when I'm walking around, if I see something which is related to trying to help students to understand physiology. So this is a picture of a gate, obviously. And obviously this white flowers, this is not Japan. It looks like cherry blossom. This represents obviously a higher concentration here and obviously much lower concentration there. So you can imagine this as the cell membrane. So for the for every living cell, there is always this very large transmembrane gradient for sodium. And so 10 times more sodium concentration outside the cell compared to the inside. So there's always this gradient, and if you, so which means that given a chance, sodium will easily move into the cell. But we talk about the movement of sodium into the cell is regulated, it is controlled, and so this is like a gate. So you can actually say that the cell is like a gated community. I don't know whether you use this word in Indonesia, where in Malaysia we have gated community, the area of houses where you have a security gate. So it's a gated community. So it's the same kind of concept that, and this gate can sometimes open for the sodium to move in by various ways. And the first one will obviously be familiar. This is the voltage gated sodium channel. So sometimes the membrane potential changes, opens the gate and sodium comes in. Okay, and this we obviously know happens during action potential. Now, the second way in which the sodium gate can open is when there is a pressure or when that specific cell is stretched. And this is obviously important in all the sensory receptors that transmits an action potential. So the sensory receptor for pressure, for touch and all that. So the pressure actually opens the gate, sodium goes in and initiate action potential. And then the third major way is that some hormone binding or even some chemical can open this gate. So we just call it ligand-gated sodium channel. So as the student goes through various areas in the organ physiology, you encounter various examples of voltage-gated sodium channel, mechanically gated sodium channels, and the ligand-gated sodium channels. But all of this point to the fact that this single gradient is already prepared. It's always there, okay, waiting to fulfill and perform a function. So depending on how the gate opens in these three different ways, once the sodium go in different cell type, it will begin to initiate different events. Okay, so let's move to. So this is one of my tagline for this major cation, that if you do not have this gradient, then many events in physiology cannot occur. So these are just examples. Some students may not have done some of the other parts of physiology, but use this as a reference. Just to mention here also that I would normally ask the lecturer in charge, Dr. Rauza in this case, to share my PowerPoint slide with all the students. So don't be in a hurry to try to copy down. Just sit back and take some notes, and then you're welcome to have this set of PowerPoint slides so that you can use it for your future reference. So let me run through this quickly. So as I said, the same gradient is seen in the sodium influx during depolarization. And then when you move to cardiovascular physiology, this same gradient is linked to the removal of calcium from the myocardium. And this is an important process whereby during relaxation, you need to lower the calcium inside the cell. So obviously the ventricle pump is a rhythmic pump. It needs to contract, it needs to relax and be topped up again with blood. And so this is obviously a very important process. And one of the transporter responsible for this is the calcium exchange with sodium. And then this is more familiar. After every meal, the reason why you can absorb your glucose and the other essential energy substrate is actually because of this secondary active transport linked to the same gradient. So without this sodium gradient, you cannot absorb the glucose, the final product of carbohydrate metabolism, as well as the amino acid. So this is an example for intestine. And then when we move on to the kidney, obviously, I think we all know that the extracellular fluid pH has to be controlled very carefully. You need to maintain the pH very carefully. And of the many integrated mechanisms to maintain pH, one of them happens in the kidney, whereby proton is secreted. And again, you see this same sodium gradient appearing again, because this transporter, basically the energy in this sodium gradient is used to secrete proton, right? So again, sodium gradient comes in. And then one last example, if for the thyroid gland to make thyroid hormone, it needs iodide in the diet. And the way in which the iodide is taken up by the thyroid endocrine cells is through a linkage again with the sodium gradient. So you have a sodium iodide co-transporter that brings the iodide in, and then iodide is then coupled to the amino acid to make the thyroid hormone. So these are just some major examples. And there's another interesting co-transporter, which is a triple co-transporter, again using the sodium gradient. So as you go through your physiology program, you find that it makes it easier to appreciate some common design and common mechanism. And the first one we want to just mention here at the start of this discussion on sodium physiology is that in every living cell, the sodium potassium ATPase, which I think all students know, establishes this sodium gradient. This sodium gradient is then exploited in all these different events in physiology across the body. Okay? So just a little bit more about this gradient. So in recent years, we've actually tried to summarize small pieces of information so that students have a visual image, and obviously that helps them to conceptualize information. So this is a triangle, obviously, and this is geometry, and there's physiology information here. So we've actually called this diagram phi geometric diagram. So we have a recent educational paper which has been published, and I'll be happy to send it to students or my colleagues who are interested. So here in this phi geometric diagram of the sodium gradient, you find that we are summarizing here. So there's always a gradient, and then the sodium influx. So obviously in one corner here is very important for many physiological processes. And then this is the one that we just mentioned, that the sodium is also involved in linking itself to various solutes. Sometimes it brings another solute in, syn-porter, like glucose and amino acid. Sometimes it exchanges with another solute, like calcium, we mentioned, and the proton. Okay, so here is just to remind students that the sodium can come in on its own, or the sodium can come in, and in doing so, it either brings in a solute or exchange with another solute. Okay, so this is the diagram. So this is a summary for why the gradient is important. Transmitted by sodium, this obviously tells you about action potential transmission. In most action potential transmission, the first phase is the sodium influx, depolarization. Transported by sodium is this here. Okay. The gradient is used, the sodium gradient is used to transport solute into and out of the cell. And then the third one is, I just titled it hydrated by sodium. And this is because if you have some basic knowledge about how water is reabsorbed, very often it follows sodium. As the sodium is reabsorbed, then there is a local osmotic gradient, and then water follows. So, and that absorption of sodium, obviously, as we saw, is also involved this sodium gradient. So as sodium moves in flux or together with other solute, then water follows. So even the water movement is indirectly affected by this sodium gradient. Okay. So let's move on. Okay, so this is I do this quite a lot in my own freehand drawing. This is to remind students that sodium concentration, as I mentioned, 10 times greater outside compared to the inside. The high sodium concentration outside the cell, ECF here and ICF here, is the main determinant of the osmolarity. So we use the symbol pi for osmolarity. So the movement of water between the inside and outside of the cell is determined mainly by the sodium concentration outside the cell, because this sodium concentration contributes to the osmolarity ECF. Okay. Now this one here, another osmolarity, is due to not sodium now, but is due to, this is in a capillary blood vessel here, and this is due to plasma protein. So some students may remember that we call this either colloid or oncotic pressure. So just to summarize here, in these two major compartments, movement of water into and outside the cell is affected by osmolarity, obviously, and this is linked to the sodium concentration. Whereas the movement of water between these two compartments across the capillary wall is not affected by sodium concentration, but is now affected by the plasma protein concentration. And so there is an important role for this oncotic or colloid osmotic pressure. Okay, so so just remember that when we talk about sodium as the major osmoactive solute, the main site of action is at the cell membrane, not at the capillary wall. Okay, so so this is just a cartoon. So we've been trying all sorts of way to, and I think for my colleague, you have the same challenge of trying to engage your students in using different ways. So this is a character that we have. This is drawn by one of my students. So we'll call this Japanese student Miss Take. Okay, and obviously this is a cat here. So because of our focus on cation. Okay, so Miss Take, if you put the two words together, sounds like the word mistake. Okay, mistake, Mistake. So this Japanese medical students, and this is in one of our paper recently, will come up with various sentences to remind students of common mistakes and misconceptions. So this is the first one, that the sodium cations have no osmotic pressure at the glomerulus. This is obviously the capillary in the kidney. So basically, we're talking about the same thing, that the osmotic pressure for sodium only applies at the cell membrane and not at the capillary. All capillaries, including the glomerular capillary. Okay, so I should show you this. This, I'm not sure whether it looks like any food in Padang. Maybe it does look like. Yeah. So obviously, every day when we enjoy food and food without salt is no fun. Okay, and this is again because of the sodium gradient. So if you've done neurophysiology of the taste sensation, you will realize that the sensation the sensation of saltiness, which makes life more pleasurable, is because of this same sodium gradient, okay, at the taste receptor in the tongue. Okay. So let's move on. Okay, let me engage the audience now, students here, by asking the question. So we said that the sodium concentration is very high outside and is very low inside the cell. It's a 10 times concentration difference. And then this gradient is generated and sustained by this sodium potassium ATPase. So the question to ask is, can we say that the outside high sodium concentration is controlled by this sodium potassium ATPase? Think about it for a while. Some of my students, when asked, tend to say, yes, it's true, since this ATPase pumps sodium out. But in reality, it is not the extracellular which is maintained by this. The answer will be correct if you change the extracellular to intracellular. So the low sodium inside the cell is maintained by this ATPase, whereas the high sodium concentration outside the cell is actually maintained by other mechanisms. So let me show it to you in this way again. This is a cartoon drawn by the same student. So Mr. K again. Mr. K is talking to, now this is another student. We'll call this student OIC. Sounds like a Japanese student. Actually, it means OIC. That means the student suddenly understand. OIC, okay, something like that. Okay. So Mr. K, tell me the sodium potassium ATPase control the extracellular. And Mr. K here says, no, it's the low intracellular sodium. And then he asks, then what regulates the outside sodium? And then this is the conversation. So let me tell you the answer here. So again, this is a Phi geometric diagram. Now we are interested in the sodium concentration outside the cell, ECF, and the sodium concentration inside the cell, intracellular fluid. Okay. So as we just said, the intracellular sodium, very low, at about 14, 15 millimole, is maintained low by the ATPase. Whereas the high extracellular sodium, about 140 millimole, is actually maintained separately by this hormone called antidiuretic hormone, or ADH. Okay. So let's move on. Okay, now, so you see, this may be the last physiometric diagram, and I hope the students can appreciate this. So we're going to move on to look at more of sodium physiology, but I want to share this quite fundamental concept, and that is the three parameters here. One is sodium concentration in one corner here, okay, represented by this sodium in this bracket. This water here represents what we call as control of water balance, okay, control of water balance. And then this OSM obviously is the osmoregulation. Now, although the words are different, obviously, but actually in terms of the physiological function, when you say osmoregulation or when you say control of water balance, it also means that you are controlling sodium concentration. So that's why the three parameters is put in the same triangle. So these three events actually it means the same thing. So when let me give you an example here. If you drink a lot of water, your osmolarity obviously goes down. You are in positive water balance and your sodium concentration drops. This all three happen at the same time. And when you get rid of the excess water, then the water balance becomes normal, your osmolarity becomes normal, and then your sodium concentration also becomes normal. So when you osmoregulate, you are actually maintaining water balance. You are also controlling the extracellular fluid sodium concentration. So this is like your coffee 3-in-1 here, because all these three actually refers to the same thing. Okay, so this is to just highlight one concept. Since sodium ECF concentration is the main determinant of ECF osmolarity, these two are associated. The osmoreceptors that some of you have really learned present in the brain, since the two are related, are actually functionally detecting changes in ECF sodium concentration. Okay, so keep in mind that this is in bold, sodium concentration, because towards the second half of this presentation, we will talk about how the body controls another parameter called sodium balance, which is different from sodium concentration control. Okay. Okay, so this is the keyword which you see in my virtual background. Yes, this is the same shell. So here you have the few keywords: regulation, control, maintaining balance, and so obviously this involves different kinds of negative feedback regulation. So just for some general education, the word homeostasis comes from this person, Walter Cannon, an American physiologist who coined this word, and to illustrate the importance of maintaining a constant environment for all the cells. But much earlier, I think in the 18th century, this French physiologist Claude Bernard already conceptualized the importance of the fact that the fluid that bathes all the cells in the body, and he calls this the internal environment, has to be kept constant. Whether it's pH, whether it's sodium, whether it's potassium, calcium, whatever, has to be maintained constant. So just a few slides. So this is Walter Cannon when he's younger, and he is also the one that gave you the word fight or flight reaction when you are reading about the sympathetic nervous system. Okay, so this is his classic book, Wisdom of the Body, meaning the ability of the cells to respond very discerningly and carefully to different kinds of challenges. Okay, the wisdom of the body. Okay, so here we are. So we looked at sodium transmembrane gradient. It's very important across almost all organs in the body. We looked at about sodium concentration control. This is mainly by the hormone ADH outside the cell. And now we're going to look at sodium balance to see the difference between these two. Okay. Now this is a little bit crowded, but there's a fair bit of information here. I'll show this slide again later on. This is just to give you this is the cat in the balance sodium. The concentration control is not the same as balance. Okay, and this is very important to differentiate. Let me give you just this is the example that so if you drink a lot of water, you become hyponatremic, okay, meaning that the sodium concentration is reduced. But if I ask you what has happened to the total amount of sodium in the body, obviously there is no change. You have not added any sodium to the body or remove any sodium. So you can have low sodium concentration but unchanged sodium balance. Okay, so it means, just one more example, if you exercise, and I hope all of us do, obviously you become dehydrated, so your sodium concentration increases, you are dehydrated. Okay, now if I ask you what has happened to the total sodium in the body, which is the meaning of sodium balance. Now obviously salt is present in your sweat. Every time you sweat, you are losing some sodium from the body. So obviously you are in negative sodium balance. Okay, so to summarize, you can have low sodium concentration and unchanged sodium balance. They are different things. You can have high sodium concentration and actually negative sodium balance. Okay, so obviously they must be talking about different things. Okay, sodium balance is total body sodium. Sodium concentration is obviously amount divided by the volume of the body fluid. Okay. So I won't go through the rest, but just to highlight those two. So again, Ms. Take says you don't need to lose sodium to become hyponatremic, right? Because so if you see a patient with low sodium concentration, not necessarily the patient has lost sodium. It could be that the patient has retained a lot of fluid in excess water balance. You can just dilute the total body sodium and become hyponatremic, right? Yep. Okay, so this is the intentionally inserted because things are going to get a bit more complicated. So we need a little bit of lighter movement. So this is one of my favorite cartoons, and occasionally it comes out with some science and biology. So this is a case of hyponatremia. And I'll leave it to you to decide whether the physiology is correct, but this is more for the human. Okay. Okay. This is one of my favorite slides, which helps us to see more clearly the difference between sodium balance control and water balance control. Now remember the triangle. Water balance is related to osmoregulation and is related to sodium concentration. That triangle. Remember. So let's look at the right side first. So on this right side, I think you don't need the word severe here. Every time you sweat, you lose some salt in the sweat. Okay, so obviously you lose water also. So this side is negative water balance, and this also means high osmolarity, right? So water balance, osmolarity, and now in purple, this will be associated with increased sodium concentration, right? The triangle, remember? sodium, water, and osmolarity. So here the mechanism is very rapid. This is basically osmoregulation. It involves just a single hormone acting together with the osmoreceptors in the hypothalamus. So obviously you reabsorb osmoreceptors here, senses the high osmolarity or senses the high sodium concentration. It releases ADH or vasopressin, and then you recover more water from the kidney. And when you recover more water from the kidney, you reduce the sodium concentration, and then you restore the water balance, and then you also reduce the the osmolarity. So this side is control of sodium concentration. This side here is now the control of sodium balance. Because whenever you lose sodium from the body, in this case it's through sweat, you need to eventually recover the sodium that is lost. And here is where the kidney comes in, okay? So this is GFR, glomerular filtration rate, and so on. Now, sodium excretion will obviously be less in response to the fact that you need to reabsorb more sodium from the kidney. So this tells you that the kidney is not just an excretory organ. The amount of sodium that you excrete in your urine, either more or less, is actually part of an homeostatic control. If you have less sodium in the body because of this sweating, the kidney will always reabsorb more sodium. And so you excrete less sodium. Okay, it always happens. So I tell my students that every time they go to the washroom, they are performing homeostatic work, because the kidney is actually, by excreting more or less sodium, indirectly trying to restore your sodium balance. Now here you notice is the word volume. The sodium, total body sodium or sodium balance is linked to blood volume or plasma volume. Okay, so the way the body does this is to increase sodium reabsorption, and one of the key hormone, if you've already done endocrinology, is this hormone aldosterone. Aldosterone will always be increased when you want to restore sodium. Okay, this is a key hormone. And then the second way in which the kidney temporarily reduces sodium excretion is to reduce the filtration, because sodium comes out with the filtrate. Okay, so this is obviously considered as a temporary homeostatic compensation. You filter less, you reabsorb more, and the combination of these two will be you excrete less when you want to recover more sodium. So okay, so these are some papers I just wanted to, in case some of you are interested, that when you want to understand physiology, you actually cannot rush through it. And of course, your curriculum is very busy, and sometimes there's not much time to process understanding of physiology. Some subjects which requires more memory work, you can do it if you have good memory quite rapidly, and most of you can do that. But for physiology, sometimes I use the word it's deceptively simple. It appears simple until the questions are asked, and then you might find some problems trying to understand it. Okay, so this is just an education paper, and this is another way in which you can help yourself to come to a better grasp of physiology, and that is to try to write out what you understand. Okay, by writing focuses the mind. So although you have your answers in your brain, but when you try to write it out and simplify, it actually helps yourself in the process of learning. Okay. So this is just another cartoon. Is life a multiple choice or the true-false test? And then the voice comes to me out of heaven and say, "We hate to tell you this, but life is a 1,000-word essay." And physiology is a 1,000-word essay. It requires words to explain and all that. Okay. So let's move on back to this one here. Okay, this is just a slide again, just to highlight there's a difference. Okay, I'm just repeating myself here. Okay, now I realize that some of you may not have done renal physiology yet, but the kidney is the key organ when you want to consider how the body maintains its total amount of salt. Obviously, we're talking about sodium chloride, okay? And this is the general equation, just to leave it with you. is, as I said, whenever the volume changes in the body, and all body fluid volume contains salt, contains sodium. So whenever you lose any volume, whether it's sweat, whether it's blood donation, whether it's diarrhea or vomiting or whatever, you enter into negative sodium balance because all body fluid contains sodium and chloride. Okay, so and as I said, how do you recover the volume? Is by recovering the sodium balance. And this is the role of the kidney. The kidney can excrete less or more sodium depending on your sodium status. Okay, so so in the case that we saw a person who loses sodium in the sweat, you need to reduce this excreted sodium. So if you want to reduce excreted sodium, you always reduce the filtered and you always increase the reabsorb. This always happens. Okay, and the two words here in red is just to summarize information that you learn later in renal physiology. One is this family of hormones called the renin. Oops, sorry. RAS is the renin, angiotensin, aldosterone system. The word aldosterone has been mentioned in the previous slide, and this is actually the hormone that is part of the renin, angiotensin, aldosterone cascade. Okay, whenever the kidney releases renin, it will eventually produce aldosterone, which will then recover or reabsorb more sodium. But the second one, which is very unusual, and this is actually a nerve, renal sympathetic nerve. So what we're saying here is that the renal sympathetic nerve is actually very important in the regulation of sodium balance as well as blood volume. Yeah. So as a sentence, this sounds a bit strange, right? That the nerve is involved in volume control. But this is true. And one of the major links is because every time you have an increased renal sympathetic nerve, you secrete renin. Because the cells that secrete renin are stimulated by the renal sympathetic nerve. So this is the link. Okay. So let me go on to the next slide. Okay. Now this is a bit complex. But you have this PowerPoint slide for your own reference. So normally, if my students are seeing this for the first time, I said, fasten your seatbelt. I'll show them this slide. Okay, now, so don't worry so much. Just look at what's happening. At the very top is decreased plasma volume or blood volume. So in terms of our focus this afternoon, how would you describe this in terms of sodium balance? It obviously be negative sodium balance. You have lost body fluid, and so you are definitely in negative sodium balance. Now, how will the body respond? Before you look at all the detail here, just look at the one right at the bottom in yellow. This is where the kidney comes in. If you are in negative sodium balance in the green part, you will always end up compensating by decreasing sodium excretion. You want to restore sodium balance so that you restore the volume. Okay, so this always happens. And how do you do this? How do you decrease sodium? Again, you see this keyword here, aldosterone secretion. Because aldosterone is the key hormone that increases sodium absorption from the kidney. And where does aldosterone come from? This is the RAAS I mentioned, renin, angiotensin, aldosterone system. Okay, so what we're saying here is that whenever you have a drop in the blood volume, renin will always be increased. It always happens. So some things are always true. Some of you who are trained to recognize false statement is that they tell you that if you see a sentence with always, it must be false. Okay, but certain things are always true, and this is one, is that the body responds to a drop in extracellular fluid volume or blood volume by always increasing renin, because it needs to restore the sodium and eventually the volume. And then this is where we come to the more complicated. There's actually a three pathway mechanism whereby renin is released whenever there's a drop in volume. Okay, one, as we just mentioned, is the renal sympathetic nerve, the RSN, in the previous slide. Okay, in red here. Okay, Now, the second two, which this is all part of renal physiology, let me just mention so that you know what this means. There is a specialized cell in the kidney, which is called the macula densa, MACD. Okay, macula densa reminds you of McDonald, macula densa. This cell is important in also helping to release renin. Okay, and then this RPP is renal perfusion pressure. Okay, renal perfusion pressure is the same meaning as renal arterial pressure, because the arterial pressure is the one that helps blood to perfuse through the kidney. Okay, so what we're saying here is that the renal sympathetic nerve direct stimulation of these cells to release renin is always accompanied by another signal from the macula densa that also stimulate the cell to release renin. And also there is a direct sensing mechanism whereby if the volume drop, blood volume drop, the blood pressure drop, which also means the renal arterial pressure drop. This is also sensed by these cells. And all three are working at the same time to release renin. Okay, so it's just to show you how important this mechanism must be, because to ensure that renin is always released, you have this triple pathway for the release. Okay, so now you're wondering what this is. So I have a sentence here. Now, if I put up this sentence, ECF sodium concentration is not regulated by aldosterone. Now, when I first put up this sentence for my students, most of them say this is true, because normally students who have done more physiology, when you look at the word sodium, you immediately think of aldosterone. So if you see these two words, you would assume that this is true. But actually, this sentence, this is a true statement. Sorry, I beg your pardon. It is not regulated by aldosterone. It is regulated by ADH. Remember when I first talked about the triangle, the sodium concentration is regulated by ADH. But if you change the concentration to balance, then it is regulated by aldosterone. There are two different things happening. Okay, so this is just to show you the person who discovered renin. And let me go to the macula densa cell. Okay, so this macula densa cell, this is in more detail for your reference. There are actually two things that the macula densa do. It also, one, it causes the release of renin, which is important in sodium balance. Now, just to put this together is that there is another separate signal from the macula densa, which is involved in the autoregulation of blood flow. So I just put this together just to summarize the two major actions of macula densa. But our focus this afternoon is the role of the macula densa in the control of sodium balance. Okay, and this involves renin, angiotensin, aldosterone. Okay, so aldosterone is involved in sodium balance, not. So where are we? Sorry. Not sodium concentration. Sodium concentration, which is linked to osmolarity, which is linked to water balance, is regulated by ADH or vasopressin. Okay. So let me, maybe I'll just skip this. And then again, this is a paper from our experience in teaching students about sodium physiology, salt physiology. There are certain major misconception that regularly occurs in almost every batch of students. And we're also seeing this across the globe when we analyze some of the common mistakes made in the international quiz. That there are certain conceptual mistakes about sodium balance and sodium concentration which are made by not only Malaysian, Indonesian, Japanese, Chinese, all over the world. They seem to have this common misperception. And so we wrote this paper about students losing balance. Okay, so happy to send this to you all. So this is just a yin-yang. So in every homeostasis, there is a balance between what natriuretic is on the opposite side. It tends to increased sodium excretion, whereas the renal sympathetic nerve together with the renin, angiotensin, aldosterone system is on the opposite side. It tends to conserve sodium balance when it is in the negative status. So since we are still in the Corona season, so I'll actually use this pun. So this sounds like sanitizers and social distancers. Okay, so the kidneys are involved very importantly in sodium physiology, salt physiology. So whenever you are in negative sodium balance, the kidneys will absorb sodium chloride. So sodium chloride is saline, so they are salinizers. And if you find yourself in positive sodium balance, okay, then the opposite will happen. Then you will have this group of hormones called natriuretic hormone, which will then promote sodium excretion. Okay, so this is what we mean by trying to put this together in a summary tagline: sodium distances, sodium is excreted. Okay. So I think I will finish off. This is one of the books that Rosa mentioned. So we encourage students to process their physiology, and of course, it's very challenging when you have other subjects like biochemistry, anatomy, so many subjects. So sometimes the curriculum does not permit enough time for you to discuss, think through, and also to hopefully enjoy more of physiology. So this is one of the things that I also do. So all of you know this song, You Raise Me Up. So this is some physiology lyrics written to this song. And those of you who know endocrine will know that this stress hormone will go up and so on. And this is your corticotrophin releasing hormone in the chorus here, and encouraging my students that I'm always behind them. Yeah, okay. So as I said, I normally leave my contact number in this last slide, and we'll be happy to continue to interact with you all in Andalas. And thank you for this opportunity to talk briefly about sodium physiology. Thank you very much. Yeah.
Okay, thank you very much, Prof. Cheng, for giving us such a very informative and interesting presentation. Now we come to question and answer session. So we welcome to all participants who have questions related to the lecture that Prof. Cheng has delivered just now. Maybe by raise hand feature in Zoom. So all participants can ask question. Okay. There is a question from Dr. Lani. So please, Dr. Lani.
Thank you, Dr. Rosa, and thank you, Professor Chang, for the outstanding explanations about physiology of cells. That is very interesting and original, everything. And I interested to ask you about a topic which has mentioned the recent interactions of genes and sodium intakes on the development of hypertension. And related to the physiology mechanism, how do you explain this could happen? Which one that is more affected by the genes mostly? It is the sympathetic controls or the renin, angiotensin, and aldosterone systems. Thank you.
Yeah, thank you, Mutia. Yeah, the basic the indirect link between sodium and blood pressure is obviously through the blood volume, because sodium, total body sodium or sodium balance, is the main determinant of the ECM and blood volume. So, and blood volume obviously affects the cardiac output and all that. Yeah. So this is the indirect link, which obviously is why patients with hypertension, they generally are recommended to restrict their salt in diet. Now, of course, the story is more complex than that, because it does involve, as Mutia said, some genetic contribution also, because they, at least in the animal model, they talk about salt sensitive hypertension, and there are categories of hypertension which are not related to salt. Yeah. So the picture is obviously more complex than that, yeah. But as a general basic what do you call that, underlying foundation of how sodium is linked to arterial blood pressure, it is through the blood volume, because sodium balance affects the ECF volume. Yeah.
Thank you, Professor.
Okay, thank you, Prof. Cheng, and the question from Dr. Lani. And maybe we can welcome to another question to the participants. Okay, maybe while waiting another questions, maybe I would like also ask question to Prof. Cheng. For example, if we do exercise such as aerobic exercise, maybe around 20 to 30 minutes. Sometimes maybe some people say that we can drink much of water after the exercise, and some others will suggest that we have to drink water slowly. So according to Prof. Cheng, so which is better to recover for the dehydration after we have exercises?
I don't know, not sure what is the reason given whether to consume water slowly or fast. The once the water enters your GI tract, they have their own way of regulating the amount of water that is reabsorbed. So I'm just thinking that the whether you drink slow or fast, the rate at which water is reabsorbed is actually controlled at the intestinal level, not at the oral level.
Okay.
So I think it doesn't really matter to me.
Yeah. Okay, Prof. Cheng. Sometimes it is better to slowly, but better slowly. Some people say it, but sometimes some of them forgot and directly drink as fast as possible. Okay, maybe we're still waiting for the question from all the participants. Maybe not only student, also maybe lecture. Yeah, if there is some question related to the topic today. Okay. Actually, our participant by now 130 participant, Prof. Cheng.
Yeah.
So so quite many students and also lecture join this lecture. Maybe because some still maybe shy or something to ask question in English. We don't know yet. Sometimes they have questions. Okay. For example, if the student want to ask question after this lecture, is it okay, Prof. Cheng?
Yes, certainly. Yeah. You're welcome to just contact me through the email or WhatsApp. I'll be happy to answer the question.
Okay, thank you very much, Prof. Cheng. Maybe since it seems that maybe some of students still shy or do not have question just now, and we hope that if there is question after reading the material from the Prof. Cheng, they will ask question to Prof. Cheng directly.
Yeah, sure.
Okay, maybe because this session already finished, maybe I have some conclusion for this lecture. Yeah, regarding 3 points. of lecture, sodium transmembrane gradient, and then sodium concentration control, and also about sodium balance regulation. For the transmembrane gradient, there is a topic about transmitted by sodium, transported sodium as sodium, and hydrated sodium. And then for the sodium concentration itself is the main determinant of the extracellular flood osmolarity and detecting by the hypothalamic osmol receptor. And then changes in extracellular volume is compensated by changes in urinary sodium excretion and mainly regulated by kidney organ. Okay, maybe since we have finished this session, I will, we have, maybe all of us can give appreciation to Prof. Cheng Helming. Thank you very much, Prof. Cheng, for this interesting lecture, and also where animation make us more interesting to understand physiology. And then maybe I will back it to the Dr. Mutia Lailani. So thank you very much, Prof. Cheng.
Thank you, Rosa.
Assalamualaikum warahmatullahi wabarakatuh.
Thank you, Dr. Rosa, and thank you, Professor Chang, and thank you to all of our participants today, all of our lecturers, all of our students who have joined us today in public lecture visiting professor with Professor Cheng Helming from University Malaya, Malaysia. Before we close our event today, let's take a picture together. Maybe please all the participants open or activate your camera. Maybe in 1, 2, and 3. Next, 1, and 2, 3. Okay. Thank you very much for all the participants today. And thank you, Professor Cheng.
Thank you. Thank you very much.
This is now our last time together in our public lecture. Professor visiting, we hope to see you in maybe in other topics. And it is very interesting to enjoy your lectures with the originalist way to deliver physiology to students so that physiology can be more enjoyable and more easy to understand. So thank you, Professor James. Thank you for all the participants. Good afternoon and see you. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much, Prof. Cheng. And all of the participants, thank you very much.
Right, thank you. Bye-bye.
Bye, Prof. Cheng. Thank you, Dr. Lani.
Thank you, Kak Rosa. Izin live, Dr. Rosa.